So it's officially 101. I guess probably the question everybody has on their mind is when are we going to get our exams back? And when are the grades going to be posted? So just a quick announcement about that. We had a few folks who were taking the exam a little later than normal. So we probably are going to hold back returning the exams until later this week. And so I'm guessing that probably won't happen until about Thursday or Friday. In terms of the exam grades, our TAs will be grading them this afternoon around 3 o'clock. And so I will be teaching, I have another class until about 5. So probably later this evening is when I'll be able to put those grades into Moodle. So just kind of keep your eyes set. So probably later this evening, or I guess if you're not on pins and needles, you can wait until tomorrow morning. And those grades should be entered into Moodle to give you a sense of where you're sitting at for this exam. So I guess I'll pause there to see if Keith has any additional announcements or if anyone has any questions.
Oh, just to thank you for your good work on the exam. Thanks a lot for that. That'll be great.
Yeah, hopefully we can get everything wrapped up by then. Like I said, we just had a few other students who need to take the exam. So we'll wait until probably, like I said, Thursday or Friday to get the copies back to you.
Sure.
I'll do I'll do what I did last time, and I'll just distribute your exams into your mailboxes. And so hopefully by Thursday, I'll have a better sense of exactly when that will be, and I'll make sure everybody knows that when their exams are available for viewing. But from the class, does anybody have any questions or concerns at this time? It's always weird to take silence as a good sign, but I feel like in this case, I guess no questions or concerns can be a good thing. But if you do have any questions or concerns about the exam or what have you, please feel free to reach out to Keith or myself, and we'll be happy to answer any of those questions. This week is kind of, I don't want to say a fun week. I'm excited about it though, because we're going to be going into a deep dive of different neurotransmitter systems that we kind of just hit upon lightly. Earlier, we were doing the introduction to CNS. And so now this is our chance to kind of do, I think, a much thorough look at some of these neuromodulatory systems. And so we'll be talking about dopamine, serotonin, histamine, and the endocannabinoids, all really fun systems that do, different things to the brain. I think maybe in the introductory to CNS, I talked about how glutamate is the gas pedal, GABA and glycine are kind of the brakes, and then everything else is kind of like the modulatory system. So if you're going to treat it like a car, it's like this. These are the other things that kind of finely tune the car and the way that they run. And you can also think of it as similar to like a stereo system and kind of tuning that very specific sound that wants to be generated. But I guess it's kind of the idea is that there's all these tunings that happen. And so we'll talk about the different subtypes of receptors, the effects that they have on tuning the system. And like I said, I think this week in particular is going to be kind of a fun set of neurotransmitters to go over. So if you're following along in your textbook, like I said, most of the materials I'm presenting here are from Goodman and Gilman. It's chapter 13 from Katzung in chapter 28. And some of these chapters are dedicated solely to these neurotransmitters, and some of them are in combination with other neurotransmitters depending on some of their functions. Because I like the structure of it. I'm using this roadmap for serotonin, but I've actually copied this roadmap for both for serotonin and histamine as well, and probably will be used for endocannabinoids as well, just because I think it does a nice job of kind of running you down like just some of the historical reference of the neurotransmitter. The chemical structure, I think, is a good jumping off point, just so we can compare and contrast different chemical structures. The synthesis, how the neurotransmitter is made, synaptic properties, and so we're kind of going small to big: synaptic properties, receptor properties, distribution in the brain, and then talking more about the physiological role of this particular neurotransmitter in the brain. Obviously, from a therapeutic approach, we're interested in disease states, so we'll briefly hit upon disease states and how those diseases are presented, how those diseases physiologically manifest. And then obviously, I think for pharmacy students, you're probably most interested in things like therapeutic approaches, ADME, and then I think today we'll wrap up talking about some adverse effects of some of the treatment therapies that are proposed for dopaminergic diseases, most focusing on Parkinson's. And I see Keith's face, so I'm wondering, hi.
Just a quick note, on how important these topics are for future lectures down the line, like when we get to the antipsychotics and antidepressants and so on. The foundation that's established with these neurotransmitters is just central to understanding what's going on in those in those categories. So good stuff, to be sure. Thank you.
Yeah, and I actually have a feeling, Keith, you'll probably chat, have something to add to my next slide, which is kind of just the history dopamine, which is this is a very brief abbreviated history. I'm talking, when I say history, it's like I'm just acknowledging that it was first identified in the human brain by Kathleen Montagu in 1957. So I think in the relative history of science, it seems kind of recent. The 50s seem to be kind of like a hotbed of science. That's when we're characterizing synaptic transmission. That's when, we're identifying neurotransmitters. Just seems to be, or, even characterizing the structure of DNA. So the 50s must have been just a great time for science. When I talk about the history of dopamine, I think one kind of really interesting historical time was in the 1980s. There was this situation in San Francisco where there were these individuals, I think there were 4 or 5 of them, that had ingested meridine, which is supposed to be MPPP. It was contaminated with MPTP. And this is metabolized by MAOB to MPP plus. And so this MPP plus actually leads to neuronal death, targeted neuronal death of the dopaminergic system in the substantia nigra and the ventral tegmental area. So it was really interesting, and we'll talk about these specific regions and kind of what they do a little later on in the lecture. But because of the selective cell death in these regions, these individuals were coming into the emergency room and essentially presenting early onset Parkinsonian types of symptoms. They had the rigidity, they had the muscle tremors and things like that. So they were treated with dopamine and dopaminergic agonists. What's really interesting about this is, again, it was kind of came out of nowhere. It took them a while to kind of figure out exactly what the mechanism was for this early onset Parkinsonian. Again, they were taking essentially these illicit drugs that had been contaminated with this MPTP. And so actually ever since that period, MPTP has kind of been, I don't want to say the gold standard, but it's definitely been the, I guess, the research method of choice for people who are studying Parkinson's disease, is because it does cause that selective death of dopaminergic neurons. And so people have been using it to essentially study the underlying mechanisms of this dopaminergic death and generate possible therapies for it. Now, what's interesting to note, though, is like, as we'll talk about a little later on in this lecture, there are a lot of genetic predisposition factors that essentially lead to Parkinson's disease. However, this, I think, is this chemical toxin generates kind of a similar overlap in the types of symptomology that you see with those genetic diseases. And now that we've had the ability to create, genetically modified animals to study, how those genes lead to that disease state, this, I think, was maybe a way that was utilized a little bit before we had that technology available. And like I said, I realized that as I introduced this, that probably Keith would have a great addition to put this into context.
Yeah, I just can't keep my mouth shut.
No, it's great.
Well, people have probably noted on our book that we call the Katzung book that Bert Katzung is the primary author there and has been for a long time, but there's some other people that contributed to that book tremendously. And one of those is Anthony Trevor. You may know it amongst the author list. And Anthony Trevor was one of the main researchers in the MPTP story, along with a medicinal chemist known as Neil Castagnoli, who later left UCSF for Virginia Tech. But anyway, I thought I would just mention there's kind of a close connection there with our textbook and this fascinating story that you're talking about with MPTP. Katie, thank you.
Yeah. And I really quickly, if you are interested in this story, okay. If you are interested in this story, the MPTP story is actually on as part of the story of the Journal of Parkinson's, and it actually kind of presents it as from the clinician's perspective so the clinicians who actually treated these individuals who had this poisoning. It really just talks about what exactly that looked like and kind of how they came to the conclusion of what this poisoning actually was. So if you're interested in this story, which is, again, you know, it's just a slice of scientific history that maybe you would otherwise not really think about, this is definitely a possibility. And I'm actually just going to copy and paste, post this in the chat right now to everyone. And so it'll make it hopefully easily accessible if you, like I said, if you are so interested in this story, because it is, like I said, a piece of scientific history worth kind of noting. Okay. So yeah, so again, this is just, it's just a sliver of kind of dopamine. I think it was first chemically synthesized in the early 1900s, but its identification in the brain didn't come until the mid 50s. And then again, the situation that arose in the 1980s, again, kind of allowed us to have a greater understanding of the role dopamine plays in the brain. So in terms of the chemical structure, again, kind of starting from the smallest part of this particular neurotransmitter and expanding into like the larger systems of the effects that it has. It's a catechol moiety linked to an ethylamine. So you can kind of see in the catechol backbone in the ethylamine to, that you see in the compare and contrast of the structure shown here. It does not easily cross the blood-brain barrier, and that is something that we're going to revisit multiple times today. You'll probably get tired of hearing about how dopamine doesn't cross the blood-brain barrier, because obviously there are certain therapy treatments that get around that particular issue. Dopamine and L-DOPA are both oxidized to form reactive oxygen species and quinones, and we're going to talk about metabolism of dopamine in later slides. And they're essentially, these quinones are going to be responsible for forming aqueducts of alpha-synuclein and are constituents of Lewy bodies that form in Parkinson's disease. And so we're not going to talk extensively about the presence of Lewy bodies in Parkinson's disease, because we're not going to be going into the pathophysiology of it per se. But these are essentially some of the metabolites of dopamine are going to be present in those Lewy bodies that are believed to alter synaptic function and be one of the reasons or behind some of the effects that we see with Parkinson's disease. Okay, so in terms of the synthesis of this particular chemical, what's interesting when we're talking about all of the neuromodulatory drugs is that kind of their synthesis can be very, I don't want to say intermingled, but you can kind of get from one to another pretty easily with a few enzymes. Just recognize that the precursors here are amino acids, and so L-phenylalanine would be the precursor here. And as we're going through kind of the steps of the synthesis, it's important to note here the tyrosine hydroxylase. This is ultimately the rate limiting step for dopamine generation. And so tyrosine can enter the brain. And so once this enters the brain, it's the presence of tyrosine hydroxylase that breaks it down into L-DOPA. And from there, you have the L-amino acid decarboxylase, or sometimes called AADC, that allows for the alteration of the L-DOPA into dopamine. And so from there, like I said, we'll talk more about the metabolism of dopamine and kind of what it's broken down to, but you'll recognize that the eventual kind of metabolites or byproducts of this are going to be the DPOC and the HVA. And we'll see a couple different kind of schematics that look very similar to this, and just recognize some of the different enzymes that are responsible for this breakdown are things like the MAO and the COMT. The reason I'm pointing those out is because in some instances, those tend to be therapeutic targets, those enzymes, again, to kind of elevate the level of dopamine and to slow the breakdown of that in the brain. Obviously, if you're slowing the breakdown, you're prolonging its presence in the synapse. And there are a couple different ways that you can kind of accomplish that. Obviously, is by inhibiting these enzymes, but then also blocking things like transport. And again, we'll talk about that when we're talking about the synaptic properties. So that was essentially the synthesis, but at the end, we kind of did hit upon the metabolism of dopamine. And so here again is another great kind of schematic showing that both the synthesis and the degradation of it. This is a little bit more in depth than what we saw on the previous slide. The green arrows with red on the outside, and I've kind of realized I should stop using green and red because, you know, if an individual's colorblind, they obviously can't differentiate between the two, but I'm just trying to draw your attention to the sites of possible therapeutic targets, which I mentioned in the previous slide. So from dopamine, you can have it break down via MAOA into dopal, and then that's going to be further broken down by LDH to DOPAC or ADH, which would be dopet. And again, the TOBAC can be broken down also into the HVA. So again, those two metabolites that we'll talk about later on will be probably that HVA and that DOPAC, or that 3,4-dihydroxyphenolic acetic acid, or the homonovinilic acid. Again, are kind of those two metabolites as dopamine is broken down. So if you're a chemistry person, then hopefully this is all just resonating with you really well. Again, I think it's nice to see where these neurotransmitters come from, where they degrade down to. This is all kind of, again, I don't want to say hypothetical, but it's obviously removed from the synapse. And so it's important to recognize that these enzymes are also going to be present in the synapse and aid in both the synthesis and metabolism of the neurotransmitter. Okay, so that's, I guess, a good transition into talking about the synaptic properties. So as we kind of increase our size and of what we're talking about, right? We're talking about, we went from an individual chemical into talking about the synapse of their property. In terms of the presynaptic, so I'm just going to pause for a second. Somebody actually asked a question. So is there a favored route between ALDH and ADH, or does Dopal go through both mechanisms equally? So if we go back to that previous slide, that's a great question. I mean, I honestly am going to say I don't think I have the answer to that, just because I'm not entirely sure about the specific enzymatic function of either of these. I'll ask Keith really quick. Keith, do you know if there's any type of preferential metabolism between ALDH and ADH for the Dopal? Do you know that?
To be honest, I don't know either. I'd have to look that up, and we'll take a look at that myself.
Yeah, I mean, it's a great question, because I'm not sure if it would maybe depend on if either of those enzymes are being upregulated by some other mechanism for any given reason. It is kind of interesting to note that there are the two routes of metabolism. So it would be interesting to know kind of what is the reasoning behind them. I wonder if do you think there's any differential expression in different regions of those two enzymes, and maybe that's why there's two routes?
That's certainly possible.
Okay. Yeah, I mean, I guess we'll both have to dig in and see if we can come up with an answer for you. Thank you so much for the student who has asked that. Yeah, and I'm keeping it, keeping this, I like to give you guys your anonymity if you don't like folks knowing you're asking questions. So I won't refer to you by name. Okay, so going back to the synaptic properties. So again, this is kind of the structure that all of these neuromodulatory drugs are going to go by, just because for me it makes sense. We're talking about the synaptic properties. I always try to think of what's happening presynaptically, what happens within the synapse, and then what happens postsynaptically as it's binding its target receptors, and then how that neurotransmitter is broken down, or how the signal is silenced. And so kind of most neurotransmitters function in that way, right? You've got presynaptic release, it diffuses across the synapse, and then it hits its target receptors, and then it either is transported into the cell or it's enzymatically degraded. And so if we look at what happens with dopamine, you can see here again, all of this schematic up here should look familiar now, because we've essentially hit upon the metabolism of dopamine several different times. You can see that dietary phenylalanine essentially is altered into tyrosine that's able to cross the blood-brain barrier, and then that is imported or transported into the cell. From there, the series of enzymes essentially can break it down into dopa and then to dopamine. And once it's converted to dopamine, it's able to essentially be loaded into vesicles via VMAT2. So VMAT2 is going to be the transporter that's expressed on these dopamine vesicles that loads and creates high concentrations of dopamines within those vesicles. Once those vesicles, once a depolarization happens in a dopaminergic neuron projection, you're going to have synaptic release. Once synaptic release happens, there are a couple of different places this dopamine can go. Obviously, most often we think of things diffusing across this synapse and hitting postsynaptic receptors. And so in that instance, you can have the D1 or D2 family of receptors expressed on postsynaptic cells. You can also have presynaptic activation of D2 receptors, so autoreceptors that you see here. And then also recognize that there are a couple different transport mechanisms as well. So, you're binding the receptors, it's going to have its downstream effect. We'll talk about those receptors and what those different receptors do to the cells in a second and following slides. But in terms of like signal termination, it's important to recognize that it can either be taken up by DAT or by the postsynaptic uptake of the OCT transporters. Then once dopamine has essentially been transported into the postsynaptic cell, you can see that there's the series of metabolizing enzymes present that converted to HVA. And then we also saw the instance of the presynaptic HVA if it's also being reuptaken into the presynaptic side. With this particular slide, something I think that might be of note to budding pharmacists and pharmacologists is that there are drugs that it can inhibit different parts of this pathway, as that are seen here. So we'll talk about the effects of blocking dopamine reuptake. Drugs like cocaine are going to target that. MAOIs are going to obviously inhibit MAO, and then alpha MPT will inhibit that tyrosine hydroxylase. Serpentine will inhibit the vesicular loading of dopamine. And so again, this slide looks, I think, I want to say fairly simple, but I think there's a lot of really interesting and good information here, because for me, we're talking about pharmacology. I think it's so important to kind of bring it back to the receptor level, to the target level, like what are these drugs actually binding to, and what are the physiological responses to them. So I think this particular slide does a really good job of showing you those receptor and transporter targets. And then hopefully from that, you can start to surmise what the effects on the dopamine levels will be, whether it'll increase dopamine in the synapse for prolonged periods of time, increasing dopamine signaling, or in some instances, if it's going to accelerate dopamine, let's say metabolism or what have you, and the results of that. Let's see, is there anything else? I guess I've kind of belabored the point that multiple drug targets exist in the system. And we'll talk about multiple drugs, obviously, a little later on in this lecture. Okay, so in terms of the receptors and kind of breaking them down into categories, I think often we talk about different categories and how they are related to one another. And so dopamine receptors are essentially categorized as like D1-like or D2-like. And then within those types, you have subtypes. So within the D1-like, you have D1 and D5. And within your D2, you have D2, D3, D4. These are categorized primarily off of off of the G proteins that they're coupled to. So we see in the description on the slide, as well as in the I guess not, it doesn't really go into the table, but in the figure below, it shows you those different G protein couples and kind of what the downstream effects of those. So for the D1 receptor family, you're going to see an activated adenylate cyclase and an increase in cyclic AMP. And then for your D2, you're going to see a decrease in cyclic AMP, an increase in potassium currents, and a decrease in voltage-gated calcium currents. And so having these different receptor subtypes, I think, really affords the brain a lot more flexibility and allows you to turn it up and turn down the signal in the presence of just a single neurotransmitter. And so it's interesting that in some instances, they almost have opposing effects. As we're looking at this slide, I am going to acknowledge the fact that I have included the receptors and then the agonists and antagonists. These are very much the names of the agonists and antagonists that would be utilized for research purposes. I'm sure they have other general names that people call them by, but I would say I'm not going to be asking you to, block D1 or D4 receptors with specific antagonists with these 3-letter names with the laundry list of numbers behind them. Although this is, pharmacology, I don't necessarily think that we need to focus on kind of that level of resolution because I think we'll be talking more about the generic drugs, not the drugs that are used for research purposes to characterize the different receptor subtypes. Okay, I've been chatting for a while. I think you can tell I'm a little excited by this material. So I'm going to pause really quick and ask if anybody has any questions. Okay. I was told that I should pause and ask for questions, whether they're questions or not, because it allows you to kind of catch up. because yeah, I do speak rapidly, so I'm trying to keep it under control. So kind of like a further deep dive, I think, into like the different subtypes of receptors. What's also nice when you're talking about receptors is to talk about their distribution in the brain. Like, you know, as we talk about them having kind of various, you know, increasing or decreasing camp levels, kind of opposing different functions. Also talking about where those receptors are expressed at, and those expression patterns are, you know, going to relate to the effects that they have on larger cognitive function. And so the D1 receptors, just recognize that that's the most highly expressed in the brain. We're not seeing it just in the brain. It's also expressed in the kidney, retina, and cardiovascular system. We'll talk about those other systems very briefly as to like what the dopamine receptors are doing in those systems. The neostriatum expresses D1, but it's not coupled to GS in that instance. It's coupled to the GOLF. And so the downstream effects are slightly different in that one region. The D2, it's the second most highly expressed. I guess just again, these are just notes that were kind of given in the textbook as to like D2S functions as a presynaptic autoreceptor. And so that's just kind of one subtype of a subtype that functions as an autoreceptor. And so sometimes the function of those autoreceptors are to either decrease or increase presynaptic release, which is something we'll talk about a lot when we talk about the cannabinoid system, just because they work as presynaptic receptors. And so we'll that's kind of a fun another aside that we'll get to. But because of its coupling, will alter that potassium, alter the N-type calcium channels and L-type calcium channels. So kind of like we had talked about before. Recognize as we're talking about dopamine receptors, we are talking about obviously metabotropic receptors. They're all G protein coupled. You're not seeing any ionotropic receptors. And somebody raised their hand. Rebecca, I'm going to pause.
Sorry, no, I just wanted to know. So these are all dopamine receptors, correct? So yes. Is like the response modulated based on how many there are, or does dopamine bind to them with different affinities?
It can be both, I guess, is kind of the answer to the question, because receptor expression is going to affect the intensity of the response. And then the affinity of the receptor might vary between subtypes, but I'm guessing it won't vary like greatly. I think receptor expression is probably a better way in which the response is modulated. And again, I always give my answers and always defer to Keith, because I think that's one of the wonders of having him here. But would you agree with that, Keith?
Yeah, that sounds right, Katie. And I would particularly like to note the D2 receptor, because it's such an important receptor as far as known antipsychotic drugs at this particular time. I'm sure that's going to change during everybody's lifetime, but currently I'm not sure I can give an example of an antipsychotic drug that doesn't interact with D2. Big deal there. But yeah, what you folks have been talking about sounds right on to me.
Yeah, I mean, but I will say, Rebecca, I love that you're thinking about affinities for receptors, because I think that that's an important point in pharmacology that, you know, maybe we're not talking enough about. But I know as much as people, you know, when talk about affinities, you're starting to obviously hit kinetics and things like that. But that's obvious. That's always going to be an important point with receptors, like how high of an affinity of a receptor is something. Like going back to the glutamate receptors, NMDA receptors actually have a much higher affinity for glutamate than AMPA does. I guess I'm kind of pulling back to that just because that's kind of my area of expertise. But I think it's definitely worth doing, again, a little bit more research and seeing if there's any difference in affinity between the receptors themselves. But I would say receptor expression probably would, you know, tone responses a little bit more. DA is an abbreviation for dopamine, yes. And then the other question is D2. So as we're going through like the different receptor subtypes, so there's D1, there's D2, there's D3, D4, and D5. So these are all different essentially subtypes of receptors. And so these different subtypes of receptors are going to be coupled to different G proteins, and that's going to essentially affect the types of response that they have. That was a question. I just want to ask the person, did that answer your question? Yes, see, so this would be D2 dopamine receptors. And yes, as I think somebody else asked me if DA stands for, what DA stands for, and DA does stand for dopamine. So Sorry. You know what? You should never put some nomenclature on your slide without defining it. DA is dopamine, so I apologize for that. Yeah. But thanks for bringing it to my attention. So I guess as we're kind of going through the laundry list of dopamine receptors, you know, as Keith was saying, you know, D2 receptors play an important role in in a specific drug class. D3 is expressed in the limbic system, as you can see here. So again, one of the reasons I included this particular graphic was because I liked that it showed the different expression patterns, so you could clearly see, you know, this subtype is expressed in this region. Although I would say the fact that the green is D1 through D5, I don't know if that necessarily helps show the differences, but I mean, showing that D1 and D2 are in that corpus striatum, and then D3 and D5 are in that hypothalamus again, which would be part of the limbic system. D4 is also expressed as, I guess, you know, close to the central nervous system in the retina hypothalamus. PFC stands for prefrontal cortex. We'll talk about the PFC in a couple slides here. So if you see PFC, I'm talking about the prefrontal cortex, the amygdala and hippocampus, again, areas near the limbic system. And this, in the case of D4, you're coupled to GIGO and activates adenylate cyclase and depresses intracellular CAMP levels. Oops. And then lastly, D5, highly expressed in the hippocampus, the area of the brain important for learning and memory, and then also found in a variety of other regions. Activates GS and GOLF and increases CAMP, modulates those sodium currents, and actually affects 3 different types of calcium channels, and has been shown to have some indirect effect on GABA signaling as well. So again, it's interesting to note that, you know, these are all dopamine receptors, but they all have slightly different expression patterns and then slightly different effects on the intracellular signaling pathways that they activate as well. Okay. Okay, so kind of again, continuing to talk a little bit about the synaptic activity. We just went through the receptors. Let's talk a little bit about on termination of the response. So the dopamine transporter, okay, label this one. DAT is the target of therapeutic and psychostimulant drugs. And so this is going to be the target for both methylphenidate and amphetamine that are and obviously at a therapeutic dose. These are used to treat things like ADD and ADHD. So when we get to talk about physiology, there's obviously a very important role of dopamine for things like attention. It's going to be crazy when we start talking about the physiology of the dopamine receptor because there's so many different functions in the brain. But back to the dopamine transporter. So it's a sodium coupled. So it basically, it's a SIM port that uses the sodium concentration to import dopamine into the cell. It's a 12 membrane spanning domain transporter. So I included kind of a picture of what that looks like, both the cartoon and in the 3-dimensional crystal structure. And I guess I kind of already alluded to the targets of the therapeutic targets of bupropion is used to treat depression and smoking cessation. So again, kind of like I alluded to just a second ago, it's amazing kind of the different different kind of cognitive order or cognitive progressions that the dopamine just entire system has a hold on. A couple different times during this lecture, I'll talk about knockout mice. Again, knockout mice are a great tool to, you know, remove a gene or remove a protein from a system and kind of see what the downstream effects are. So there are dopamine transporter knockout mice that show to be hyperactive. So again, that role it has in attention hyperactivity, and do not display increased locomotion when given cocaine or amphetamine. So this result probably isn't too shocking given the fact that the target of cocaine and amphetamine are going to be the transporter. So in absence of the transporter, you're no longer seeing the effects of those drugs in that system. So again, this kind of one of the wonderful uses for knockout animals is to actually study the underlying effects of these drugs. So somebody popped up a question, and I'm just going to read it out loud because I think I'm going to need a little clarification here. So we learned that mice can now be humanized. Are these the kind of mice, the ones used in knockout mice? So the humanization, I guess the humanization is in what regard? Because that's I guess my question. I haven't heard that term humanization.
So in like medchem, we were learning about antibody drugs and that they were created in mice and the mice were humanized, so you didn't have to change it or something like that.
Okay.
That's
Oh, okay. So yeah. So in that instance, so it would depend. I guess to humanize a mouse, so a knockout mouse would not be humanized per se because you're taking the endogenous gene and you're removing it from that mouse. So it would be the endogenous dopamine transporter that you're removing from that mouse. Now, it might be considered a form of humanization if, kind of in a reverse way, you decided to increase expression or exogenously, like let's say inject a virus into a mouse and have them express the human dopamine transporter. So that would probably fall on the lines of humanization because you would be having them express the human form of the gene in that system. But you might, if you're interested in studying the function of the dopamine transporter, you would probably have to couple a dopamine knockout mouse with a human dopamine expressing mouse. Does that make sense? So you could study, you could basically clear out your background and get rid of any type of dopamine transporter that the mouse would normally be expressing, and then express the human form of the dopamine transporter. And I guess the only reason to do that would be if you were interested in the differences between the two, which I'm guessing would for the purposes of people who use mice as research models would hopefully be negligible. But I think if you were being a very careful researcher, you could go about that. I hope that, does that answer your question in any way?
Yeah, it did. Thank you very much.
Okay. Yeah. I mean, and it's and with how science is going, it's amazing what we're able to accomplish in terms of really picking apart individual components of a system where you're able to literally knock out a single gene or like recreate SNPs in animals and just really get down to the fine nitty-gritty details of exactly what a residue on a protein is doing in an animal and their ability to process, let's say in the case of the dopamine transporter. Okay, I'm devolving. I need to stay on task. Okay, so I hit upon, obviously we're focusing a lot on the central nervous system. No, don't apologize. It's great. It's a great question. And I get chatty, so it's my fault. So the distribution in the body, as I said before, we're talking primarily about central nervous system effects, but it's worth noting that it is distributed in the cardiovascular system, the kidneys, the pituitary gland, and then I just kind of threw in the central nervous projections here at the bottom, and we'll obviously have another slide dedicated to just CNS. But in the cardiovascular system, the D1 cause vasodilation and can reduce cardiac load. And so at high concentrations, dopamine activates the alpha adrenergic receptors and can cause vasoconstriction. So this is used in severe congestive heart failure, sepsis, and cardiogenic shock. The treatment of this is administered via IV but it's generally not chronically administered. It's generally a short-term administration for these particular disease states. In the kidneys, there's probably a little bit longer of an explanation in your textbook, talks about the paracrine autocrine transmitters to D1 and D2. They're going to regulate sodium transporter, and then D1 increases renin secretion and D3 reduces renin secretion. Again, that's not surprising based upon how they're coupled to different G proteins. The pituitary gland, it regulates prolactin, bromocriptine, and caboglobin are used to treat hyperprolactinemia. And I have hyperlinked the kind of the NIH kind of rundown of this particular disease state. So if you're interested in it, by all means, go ahead and open the PowerPoint and click on this, and you can read all about this disease. Administering dopamine, yes. In that case, it would be administering dopamine. And since you're administering it into the peripheral system, you could administer straight dopamine. You don't have to worry about it crossing the blood-brain barrier. So the question, I guess, that somebody popped up was, are they administering dopamine? I'm assuming for cardiovascular effects, and the answer is yes in that instance they are. Okay. Good questions today. Any other questions before we proceed? Okay. Oof, okay. So for the central nervous system, there are three major projections that are important for dopamine to be distributed to the brain. You've got your mesocortical mesolimbic, your nigrostriatal, and your tuboinfundibular. Kind of a fun word to say. This particular image, again, is straight out of your textbook, and I think does a nice job of just showing these projections. Again, you can see that the origin of nuclei for dopaminergic cells are located in the substantia nigra, and then you have those projections extending down out into the frontal cortex. Again, we'll talk about the effects of dopamine on the prefrontal cortex. You can see that the nigrostriatal pathway is the upward dashed blue line that's located here. The mesocortical mesolimbic is going to be the solid blue arrows you see projecting into the cingulate gyrus, as well as the striatum. And the tuboinfundibular pathway is going to be this red pathway that you see down here. And again, we just kind of just very quickly mentioned the effects that dopamine has on the pituitary gland. So it's going to be this projection to the pituitary gland that's going to be releasing it to that region. So again, it's just these three different pathways are obviously having different effects. We'll talk about those different effects. Again, it's one of the amazing things about dopamine is all of the different cognitive outcomes that it essentially causes. Okay, so as we're talking about some of the physiological functions of dopamine, it's important to recognize that there are movement disorders that are related to the lack of dopamine in the system. So one of the functions of dopamine is going to be to mediate movement in a smooth way. So within movement disorders that occur in the disease states, you can see that the circuitry in which we kind of just looked at gets disrupted. And so in this case, the circuitry of the cortex, basal ganglia, the thalamus, leads to an overactivity or this red arrow pathway that you see here from the striatum to the globus pallidus. And so when we're looking at some of these movement disorders, the way that they're going to present are as tremors, chorea, and tics. And so you can kind of see the different kind of characterizations here of each one of those. You recognize that many of these symptoms that we're looking at here probably overlap with maybe some of the symptoms that we'll talk about when we get to Parkinson's disease. So the tremor will be the rhythmic oscillatory movement around a joint. Chorea will be the irregular, unpredictable, involuntary jerks, and tics will be sudden, coordinated, abnormal movements that tend to occur repetitively. And again, usually the mediation of these movements would be conducted by the dopaminergic pathways that are intact. And then if those dopaminergic pathways are no longer present, you see these symptoms begin to present themselves because you don't have dopamine mediating the fluidity of these movements. Okay. So in terms of kind of looking at the effects that dopamine has, or some of the physiological functions, again, it's amazing the variety here. So it obviously has an important role in reward, emotion, cognition, memory, and motor activity. So that's, again, spanning a lot of different functions of the brain. This figure here is great because it shows you some of the dopaminergic pathways, some of which we just talked about. And then tomorrow we'll continue our discussion and talk about serotonergic pathways. But again, the functions: reward, pleasure, euphoria, motor function, compulsion, preservation. Dopamine is so intertwined with motivation and our reward systems that an argument could be made that a majority of our decision making is based off of dopamine release. In the study of D1 and D2 receptors, we talked about the function of knockout mice a few slides ago. So when scientists are studying the functions of these specific receptors, they've noticed that in D1 knockout mouse, there's a reduced reward for ethanol, so they no longer have the seeking behavior. They've also recorded that in rats, monkeys, and humans, that too much or too little D1 stimulation can impair the prefrontal cortex. And so it kind of has this U shape, obviously this perfect amount, not too much, not too little, everything in moderation kind of thing. So it's got this U-shape effect of dopamine on prefrontal cortex function. So if you have too much dopamine, it's not a great thing. If you have too little, it's not a great thing. So in order to preserve that prefrontal cortex, which is important for higher level cognitive thinking. D2 receptors in those knockout mice, they also show reduced reward for ethanol, reduced reward for morphine, and then enhanced self-administration of cocaine, which is kind of interesting that they're observing that as well. Again, the target of that is going to be the DAT, or the transporter system, and not necessarily the receptors. In terms of alterations in signaling, we'll kind of expand upon one or two of these disease states. Can lead to PD, which is Parkinson's disease, Tourette syndrome, bipolar depression, schizophrenia, ADHD, and attention or substance abuse. So it really does kind of span the gamut of kind of cognitive disorders, addictive disorders, neurodegenerative disorders. And so it kind of just, again, really hits home about how important dopamine is to brain function, and how a dysregulation it can lead to a variety of different disease states. Oh goodness. Okay, so focusing in on Parkinson's, if we look at Parkinson's kind of as, I don't want to say the prototypical dopaminergic disease, but the fact that it is presented by specific loss of dopaminergic neurons. It does give rise to a lot of the different physiological functions that we just saw. So the motor control is probably one of the most apparent presentations of Parkinson's disease. So you have things like rigidity, bradykinesia, tremor, and postural instability. Not surprisingly, there are also affective disorders related to Parkinson's disease, things like anxiety, depression. And then because of effects in things like prefrontal cortex, you have confusion, cognitive impairments, changes in apathy or fatigue, and abnormalities in autonomic function. In terms of Parkinson's disease, currently there is no cure, but the pharmacological treatments that we are going to talk about will slow the progression of this disease. And so the first few years, it shows to be fairly effective in mitigating some of the symptoms that we're talking about, but it's by no means a cure. And I think we're still in the early stage of understanding some of the genetic predispositions they might have. Okay. Yes. So so you are correct. Some of the images for this presentation are not presented on the Moodle version, but I'm going to upload this as soon as we finish lecturing. Just because this is essentially my version, I wanted to give you the full version of this that I uploaded yesterday, so folks would have a chance to look at it. The content has not changed a ton. There's just a few more figures, and so as soon as we wrap up class here, I'll upload this one, and it has the answers to the concept check, which I don't know if we'll even get to today, included in it. Okay. So in terms of the pathogenesis of Parkinson's disease, again, high concentrations of dopamine is essentially the normal in the brain from the neurons of the substantia nigra. So you can see here kind of the normal brain here showing that projection from the substantia nigra to the corpus striatum. And again, it's going to affect the synapses between the acetylcholine receptors and GABA receptors. And so this is important to note because there are some therapies that focus on this particular pathway here. So this impaired, the result of Parkinson's is essentially the result of impaired degradation of proteins, intracellular protein accumulation and aggregation. These are all reasons that we might be seeing that selective dopaminergic death. So it does lead to mitochondrial damage, inflammatory cascades, and apoptosis, again, of these dopaminergic cells. In terms of some of the genetic factors that are responsible for Parkinson's, you can see here that there isn't necessarily just a single gene, a single locus that's responsible for the development of this disease. There's actually several. So it could be mutation in an alpha-synuclein gene, or a mutation in leucine-rich repeater kinase, the LRRK gene. Parkin gene has also been shown to progress. And so as we're looking at these genes, they've been isolated in humans, and then they've also been generated in a mouse model as well for individuals to study the progression of these diseases that all kind of develop very similar Parkinsonian pathogenesis that we are observing here. Interesting to note that some of the things that are protective against Parkinson's, things like smoking, coffee, anti-inflammatory drugs, high serum of uric acid, and then there's an increased incidence in things like teaching, healthcare, and farming. So I'm just like, doesn't look good for me. It's just interesting to know that there are these occupations that have an increased predisposition. Um, so in terms of therapeutic approaches, where are we at? We're at 150. Oh, okay. So I think I'm gonna have to stop there. I think tomorrow we'll have to kick off and we'll have to start with, just a quick review of Parkinson's disease and hit the therapeutic approaches. I know that this is actually where I think it gets kind of good for you guys because you get to learn about the drugs that you'd be using for Parkinson's disease and other dopaminergic diseases. And I obviously can't skip it or rush it. So I guess this is where we'll kick off tomorrow. I will upload the presentation that you're viewing right now, literally as soon as we hang up from our Zoom call. Does anybody have any questions or concerns before I let you go? Like I said, exam grades should be going in later this evening.
Just a quick note, if you go back a few slides to the idea about reward. I don't know if there's a more important topic that you covered than that one, and the tremendous importance of reward phenomena in a variety of physiological and pharmacological settings. So that will come up again and again and again. Have a good day, everybody.
Yeah, thank you so much for everyone for your time. And if you have any questions or concerns about class, feel free to reach out to either Keith or myself. See ya.
Thank you.
Thank you. Katie, I was actually just going to share an interesting story about the reward in rats and stuff. So like they did that study years ago where they found that rats and mice would like press the button over and over again to get the drugs. And so if somebody did a follow-up study where they actually put the animals in like proper care environments with social and mental enrichments outside of the drugs and stuff, and they found like it definitely reduced the amount that became addicted to the drug. So I just thought that was really interesting because it kind of puts in like environmental protective factors versus like actions and stuff. So I thought that was interesting.
Yeah, I think there's a lot to be said for having a support system, like it's, man, during COVID, what a concept, right?
Right.
Yeah.
Yeah, that was a really interesting behavioral look at it. So that was cool. But
yeah, I think
blackouts.
yeah, I think we think of so often as mice just being these animals that, are not social, but they so are, like, I don't think that's often taken into account when we do these types of studies, because if you do any kind of behavioral studies on animals, usually they do them in seclusion, because they don't want to have any confounding factors.
Yes, no, I had pet rats, so I got really interested into like the social behaviors of them. And they did studies where they found that rats actually demonstrate generosity, and like they